Hey guys, so the title of this unit is protein synthesis. To really know how proteins are made, we need to go back and start with DNA. So if you'll remember, right, let's review it. Sugar in DNA is deoxyribose. A goes with T, G goes with C, and it's double-stranded. Now we're going to talk about RNA during protein synthesis as well. You guys remember there's some pretty key differences between DNA and RNA. So the sugar is different. Right? RNA has ribose. Okay, and now instead of T's, we have no T's in RNA. A goes with U. Okay, so that's really important. A goes to U. G still goes with C, so we don't have to worry about a change there, but A and U. And also another key difference is that RNA is only a single strand. All right, so again, just kind of to show you, if this is our original DNA strand, ATT, GCA, if we wanted to make an RNA strand that complemented with that, A would go to U, T still goes to A. Okay, so don't get that twisted. T still goes to A, T still goes to A, G still goes to C, C still goes to G, A still goes to U. Excuse me, that's new. A goes to U. So why does RNA matter? Like, who cares about it? Without RNA, we would not be able to make proteins, which really, remember, that's what we're focusing on in this unit, is protein synthesis, is making proteins. Okay? So, without RNA, we cannot create a code for our proteins. All right, there are 3 different types of RNA. The first one is called messenger. We're usually just going to call it mRNA. Now, remember, okay, whatever goes in front of the RNA, that letter is always going to be lowercase. So mRNA, and what it does is copies DNA. So it literally basically takes a photo of DNA. It copies DNA. Remember, DNA is in the nucleus. So in your reading yesterday, you learned, right, that something has to leave the nucleus with that DNA code. What's leaving is mRNA. The second type is called tRNA, or transfer RNA. And what tRNA does is picks up amino acids. You guys remember amino acids are the building block of proteins. tRNA picks up amino acids and takes them to the ribosome. Now remember, proteins get made in the ribosome. Y'all don't forget that from last unit, those cell parts. Now, in the ribosome, there is a piece called rRNA. Okay, ribosomal RNA. It literally sits in the ribosome, waits for tRNA to bring in those amino acids, links them up, and creates a protein. All right, so protein synthesis, or the making of proteins, happens in 2 steps. We just kind of covered them a little bit by talking about those 3 types. But the 2 steps that actually occur are called transcription and translation. We're going to go over each one. So transcription is when mRNA copies that DNA. It's important to note that this happens in the nucleus. Okay, so mRNA copies DNA, happens in the nucleus. Okay, so we can practice that. This is our DNA that's sitting in the nucleus, ACGTGA. mRNA is going to copy that, and remember it's a type of RNA, so it's going to have U's. So we're just going to match up the pairs. A goes to U, G goes to C still, C still goes to G, remember T still goes to A, G still to C, and A goes to U. That's all you got to do in transcription, is just literally copy. Okay, just changing those over. Now, any of those sets of 3 letters on mRNA, it's going to be called a codon. Okay, now a codon is what we need to make our proteins. Okay, so 3 bases on mRNA is what helps us make proteins. Now there's a rule. 1 codon codes for 1 amino acid, and 1 codon has 3 nucleotides. Make sure you write down this rule right here. It will be a quiz question. It will be a test question. Hang on, I have to go punch my brother because he's being loud. Okay, this slide looks hairy. It's not. All you need to know for translation is that in translation, we change the mRNA codons into amino acids. Okay, we're going to use something called a codon chart. You'll get one here in a minute. But all you need to write down is that we're translating mRNA into amino acids, and that eventually becomes a protein. Again, we need to write down where it happens. It happens in the cytoplasm and the ribosome. All right, so you've got a section under translation that says, how does it work? Basically, right, we just talked about it. mRNA leaves the nucleus, goes to the ribosome. Wow, that's spelled wrong. Ribosome. tRNA is going to pick up amino acids, take them to the ribosome. Our RNA links them up. There's translation. All right, so translation, we've got our DNA, right? TAC, CTA. Then we're going to transcribe to mRNA. T goes to A to U, C to G, C to G, T to A to U. Then we're going to translate that mRNA into amino acids. Don't even look at tRNA. Do not even look at this tRNA strand right now. Okay, so we translate mRNA to amino acids, and you're going to do that using a codon chart. I'm going to give you one of those here in just a second. Now, tRNA, okay, carries this cool thing called an anticodon. Basically, all you need to know about an anticodon is that it matches the codon. Okay, it matches the codon. It's basically just another RNA strand. So, A to U, U to A, G to C, G to C, A to U, U to A. Basically, if you look up here, it's almost the exact same as this DNA, right? Somebody's trying to come in my room. It's almost the exact same, apart from it has U's instead of T's. Now, your hint is that you use the codon chart, okay, and also use mRNA to find that codon chart. Now, we're going to practice. You're going to have a codon chart, and we'll figure this out together. Okay? Good luck. We'll see you later. Oh my God, I'm going to kill whoever is knocking on my door.